Okay, hello everyone. Welcome to today's webinar. I am Lily Jiang, the host today. I'm honored to have such a great opportunity to discuss with you all an extensively noted topic, the current research and overcoming challenges of antibody-drug conjugates, known as ADCs. The cancer is one of the leading causes of death. According to the latest report of WHO, cancer accounting for nearly 1 in 6 deaths worldwide. So antibody-drug conjugates are a relatively new class of anti-cancer drug with high specificity and potent killing properties. ADCs consists of 3 key components: a monoclonal antibody, a linker, and a payload. With a specific antibody targeting the surface antigen of tumor cells, ADC is internalized and processed by the endo-lysosomal system to release the potent cytotoxic drug. Each of these major components in the development of ADCs may affect the efficacy and safety. In addition, others such as internalization rate, choice of linkers, and conjugation methods may also enable a successful ADC. In this webinar, QSABIO will discuss current strategies and the recent development of ADCs, challenges in ADCs development, as well as the future directions of next generation ADCs. The speaker of today's webinar is Dr. Yin Zhou. Dr. Zhou is a product manager at QSABIO and in charge of product marketing, development, and optimization. Before joining QSABIO, Dr. Zhou obtained her PhD degree in biology and biotechnology from Worcester Polytechnic Institute in the US. After graduation, she did her postdoctoral training at the University of California, Davis. Dr. Zhou's expertise lies in molecular biology and protein science, specifically in mRNA expression and regulation, with interest in mRNA stability and RNA interacting proteins. The webinar will last for an hour, including a 40 minutes presentation and 20 minutes Q&A. So please feel free to post any questions in the QA section or the chat box. We will answer them after the presentation. Now, let's hand over the time to Dr. Zhou. Hopefully, we will spend a scholarly exchange time filled with insights and inspiration together. Thank you.
Hello, everyone. Welcome to this webinar, and then thank you for all your time. My name is Angel, and I'm a product manager in QSABIO. I'm really happy to have this chance to meet you in this webinar and share knowledge about antibody-drug conjugates with you. Please feel free to ask any questions and then share ideas with us. And I'm going to share my presentation today. So my topic today is about antibody-drug conjugates, the current research and overcoming challenges. As a new class of anti-tumor medicine, ADC provides a means to reduce the off-tumor toxicity by limiting the payload exposure in normal tissues. So according to a new analysis from the Data M Intelligence, the global ADC market size was valued at $5 billion in 2022, and then predicted to reach the 6.5 billion by 2030. And here in the right hand, I also showed a global sales prediction about current approved ADCs from 2020 to 2026. And I want to point out this blue line here. This blue line represents a drug called Enhertu, and then we usually call it TDXD. It comes from the AstraZeneca and the Daiichi Sankyo, and it have a predicted sales of more than 6 billion by 2026. And another pink line that I'm pointing here is the ADC drug Padcev. It's from the Astellas and the Seon, and it also have a predicted sales of more than 3 billion by 2026. So not only the approved ADC, the clinical evaluation about ADC also increased a lot. Here is a summary about the clinical trials. So the number is almost 1,200. It's covering the 267 ADCs. So they have been registered since 1997 to evaluate the safety and efficacies. And hopefully they will be prominent in the oncology landscape. So I already talked about the sales market, about ADC drugs. Then what is the ADC? I will explain it from the structure and then the mechanism of the action. So the ADC comprising of a tumor antigen specific molecular antibody, and it conjugated to a potent cytotoxin via a stable chemical linkers. So binding of this big molecular here, the molecular antibody to the target antigen will result this ADC complex internalizing into the cells. So after this big complex internalizing into the cells, inside the cells, the antibody portion will be degraded into the through the lysosomal degradation, and the payload portion will be released. And part of the payload will interact with the target, resulting in the cell death. And another part of the portion will taken up by the neighboring cells. So this process called the bystander effect. So with the bystander effect, the cells surrounding the target cancer cells will also be killed. So the success of the ADC in inducing this strong cytotoxicity depends on various factors, such as the characteristics of the target antigen, selecting a specific antibody, engineering a stable linker, as well as the conjugation of a potent payload. So before I talk more about the common challenge of the ADC, I want to emphasize on the history and the development of some approval ADC. So here is the timeline displaying some important events in the development of approval ADC drugs over the last century. So back to the 1910, the magic bullet was proposed by Paul. So he is a scientist from Germany. He identifies that the antibody target themselves without harming the organisms. And he also anticipates that attaching a toxin to this antibody will improve their therapeutic specificity. And then during the long history, with the development of the molecular antibody, and later the chimeric or cumularized molecular antibody advancement, in 2000, the first ADC drug called Melotac from Pfizer was approved by the US FDA. And this drug was for adults with acute myeloid leukemia. With this drug, it marked the beginning of the ADC era of the cancer target therapy. So after 2000, during the last more than 20 years, there are more than 10 different ADC drugs come to the market. So basically, we can separate the ADC drugs into 3 different generations. So for the first generation ADC, we saw that a mirroring antibody against the target antigen was used. And then the drawback of this mirroring antibody was clear. It showed a strong immune response, and also the efficacy of the treatment was not that good. And then we are glad to see that from the second generation to the third generation, the mirroring antibody was converted into a mouse humanized chimeric antibody. Or for the third generation, we saw the fully humanized antibody. So the strong immune response was reduced. And I also want to emphasize that there's another advanced technology about the linkers and the conjugation method. So with these 2 advanced development, we saw that the safety of the ADC drug increased a lot. It because that these 2 can determine an important quality attribute factor of ADC, which we call the DAR. So DAR is a ratio between the drug and antibody. And then it means that for each antibody, how many drugs that this antibody can carry. So it's kind of a representative that how better or the efficacy of this drug and how safety is this drug. We are glad to see that for the 3 different generations, the drug efficacy increased, and then with the stable linker and advanced conjugation method, the safety of the drug is also increased. And then for the 3rd generation of the ADC, we see like a great improvement in the stability and as well as the drug release rate. So the overall therapeutic window is getting larger without causing some additional whole system toxicity. And then to have a better understanding of ADC, here I showed some approved ADCs that can come from 2 different categories. So the top is from the hematological cancers, and at the bottom, they are for the solid tumors. I'm not going to introduce all, but I will focus on 3 different ADC from the 3rd generation. So the first one is the Playback. Is from the Genotech. And then for this drug, I want to say that the antigen is pretty novel. It targets the CD79B. And then the linker here is a diapeptide cleavable linker. But the payload is not that novel. It's a very conventional payload called the MMAE from the myotubulin inhibitors. And the next drug is the Padcev. So for the Padcev, comparing to the previous Playback ADC, the linker is the same, is also diapeptide linker, and then the payload is also the same, is MMAE. But the good thing is that we also saw a novel target antigen was used, so the target is the Nectin-4. And then the 3rd drug I want to emphasize is the Enhertu, which is the T-DXd. So for this one, we saw some advanced improvement. So the first is the DAR. We saw the DAR is 8, which means that for this antibody, they carries 8 molecules of DXd. And also the linker here, instead of using the dipeptide linker, this one is the tetrapeptide linker. And another thing about this amazing ADC is that the DXd, the payload, is pretty novel, and then it's very kind of like a strong. The release of this payload will increase a lot in the bystander effect. So which means that the efficacy is improved a lot with this drug. So for all these 3 drugs, we saw a similarity and then also some difference. So we're glad to see that the expanding of the target of the antigens. And then we also see advanced development of the linker technology, as well as the novel payload were chosen for the ADC. So I hope by now, everyone already have a general idea of ADC. So from here, I'm going to talk about the common challenges in the development of ADC. So here I list some target antigens that in the approved ADC, and then some ADC target that from the clinical development. So HER2 here, I want to mention that because it's a successful commercial target antigen that people commonly chose, because it has high expression in the tumor cells, but the minimal expression in the healthy cells. So one example about the expression of HER2 is that it's 100 times overexpression in the tumor cells comparing to the healthy cells. And also it has a sufficient density and homogeneity on the surface of the tumor cells. So it's accepted that ADC efficacy usually depends on the efficient target-mediated internalization. So the internalization is a particular important property of the ADC target antigens. So right now, if the ADC drug that you develop, the antigen is not in, the antigen didn't have this important property, then maybe you need to change to another one for current drugs. However, not every so antigen has this property, so it's better to determine it before you choose that one. And then after the antigen, we better to select a specific antibody with high binding affinity to this target antigen, because the high binding affinity, for some extent, it means that a repeat internalization. However, this is not always true. So antibody with the high antigen affinity may in turn reduce the penetration into the solid tumors. It's because that it just always complex in the solid tumor treatment because of the binding site barrier in here. So the extremely strong binding between the antibody and the antigen may result the ADC gets trapped in the blood vessel. And then the ADC is hard to penetrate to the tumor cells away from the blood vessel. And also the experimental result shows that the antibody with the moderate affinity can have a highest levels of the drug calculation. However, the high affinity have a low level of drug accumulation. So it's kind of like indicates that for the solid tumor treatment, the bystander effect is more important to increase the efficacy. And then for the next two, I want to say that the antibody size needs also to be optimal. So not too large, not too small. Large means that this antibody will be affected with the penetration. And then if the antibody is too small, then it may have a shorter half-life. And then the cumularized antibody I mentioned before for the third generation with less, you can definitely get a significant reduced immunogenicity for the ADC drugs. So after the antigen and the antibody, I will talk about the common challenge that related to the linker and the conjugation. So for the linker is tether the antibody to the cytotoxic drugs. And then the purpose of this one is to maintain the complex stability in the blood circulation. So you can think that the linker as a critical safeguard that can maintain the stability and then also to eliminate the excess toxicity before the internalization. So here I list some types of the linkers. So basically we can separate them into two categories. So the cleavable linker and the non-cleavable linker. For the cleavable linker, the linker can be degraded chemically or enzymatically. For the chemical linker, one example is the acid-sensitive linker. So for this type of the linker, they are usually unstable in the acid condition. And then for the enzymatically degraded linker, one example is the peptide-based linker. So for the previous, the tetrapeptide or dipeptide linker, they both belong to this category. For this type of the linker, they're usually sensitive to the lysosomal protease, which are generally overexpressed in the tumor cells. So it's easy for them to get degraded inside the tumor cells comparing to the healthy cells to maintain some extent stability of the drug. And then for the non-cleavable linker, it's different from the cleavable because the stability of the linker is pretty strong. So the drugs can after the drug internalized into the cells, they can be completely degraded to release the toxic payload. So one example about this one is the MCC linker. So this linker has been used in the ADC drug TDM1. So the benefit for this type of the linker is that increase the plasma stability and then reduce the likelihood of the premature payload release. So this can provide a greater tolerance comparing to the cleavable linker that used in the ADC. And then for the conjugation method, they are also kind of like advancement from the previous random conjugation with the naturally occurring amino acid to the site-specific conjugation. So for this type of the random conjugation, usually in the early ADC drugs, they chose the lysine, the pre-existing lysine to do it. It's because that for the lysine is a vessel method to do the bioconjugation because their natural abundance is pretty high, and then they are easy to access because they are on the surface of the side chain. However, because the random coupling with the lysine residue, the DAR is not that consistent, means that for this antibody, the number of the molecules that attached to this antibody is not that consistent. So resulting that ADC drug become like a heterogeneous in the quality. And then also the coupling reaction near the antibody and the antigen recognition site reduce the ADC binding to the target. And then this can be improved by the site-specific conjugation. So for this category of the conjugation, it have the engineering reactive system residue. And then also you can introduce of the non-natural amino acid as well as the glycan conjugation. So with the site-specific conjugation method, you can have a site-specific ADC to have a very consistent DAR than the random conjugation method. And then for this part, I want to mention a technology, the CLMAP technology developed by the Genotech. So they improved the genetic engineering technology to insert the cysteine residue at the specific position to do it. So it's pretty kind of like a popular technology. However, there are also some drawback about this conjugation method. So one example is the immunogenicity, because you introduce some unnatural amino acid, so the immunogenicity can happen. And also because you may modify the amino acid sequence, the hydrophobicity of the ADC can change. So if the ADC drug is too hydrophobic, the ADC will aggregation together to cause them to be cleaned up quickly. And then the next challenge about the ADC development is the biological bullet, the payload. So we expect the payload to be stable and then potent. Some studies before made assumption. So this assumption is based on that researchers assume that each step during the ADC release is 50%. So when they say every step means that the step like the antigen binding to the antibody, the internalization step, the payload release step. So for each step, if the efficiency is just 50%, finally, there are less than 2% of the ADC could reach the target tumor site. So then we will say that we expect that even with this small population, like 2% ADC target the tumor site, we still want them to potent enough to complete their task. So here I list some of the cytotoxic payloads that commonly see in the ADC drugs. So one is the tubulin inhibitors. So MMAE, I mentioned before, they are commonly used. So for this type of the payload, they bind to the tubulin and then blocking the polymerization, causing the cell death. And then for the next category is the DNA damage agents. One example is the PBD dimers. So PBD is a class of anti-tumor antibiotic. It was discovered back to the 1960s. So when the PBD work as a dimer, they cross-link in the DNA and then can block the cell division. So the PBD dimer is less visible to DNA repair, then it can decrease the possibilities of the drug resistance. And another novel payload that become popular is the topoisomerase inhibitors. So one good example is the DXD. So for DXD, they have a higher activity, and they also have another amazing feature about the membrane permeable, which means that with this feature, they can increase the bystander effects and then to make the drug to be more potent. And then I'm glad to say that our CursorBio have this DXD antibody with 2 different clones. So if you need to know more or you want to ask for a free trial, please contact us for some further information. We are glad to help you. And then the last type of the payload, I want to say that maybe it's not in the current approved drugs, but it's still in the clinical trial, is the immunomodulators. So for this type of the payload, we want to combine the chemotherapy with the immunotherapy to have a combination of the precise antibody target to increase the power to make the efficacy more better. And for the last common challenge I want to mention in the development of ADC, people may not think that is as important as the others, but actually it's very important. Here I showed a mechanism of action of internalization ADC. So after the ADC is internalized, only with the degradation with the late endosome and the lysosome, the payload can be fully released. So actually, to some extent, you can think that the internalization rate is directly related with the efficient payload release. And actually, most the current ADCs, they are designed to against a target antigen that display the efficient internalization, because then with this, you can facilitate the ADC entry upon the recognition. So the antibody internalization efficiency is very important. And this efficiency actually varies from antibody to antibody. And even you select antigen, when you choose different antibodies, each pair between the antibody and the antigen, they can be different. So which means that we need to assess the efficiency experimentally. And inefficient internalization will lead to an off-target release of the ADC and then result in systematic toxicity. And then to solve this issue, our Castle Bio have a recombinant protein DT3C to facilitate the development of ADC. With this efficient tool, you can assess the internalization efficiency in vitro. So what is DT3C? It contains a diphtheria toxin without receptor binding domain, and then fused with the 3C domains of the Streptococcus protein G, because the Streptococcus protein G have a Fc binding domain. So the recombinant protein DT3C can bind to the antibody and then form the immunotoxin. So with the immunotoxin, they can be internalized after they recognize a surface molecule expressed on the cells. Then after internalization, the DT3C will be cleaved by the furin protease. And then the catalytic domain of the DT3C is released. The release of the catalytic domain will lead to ADP ribosylation of the EF2, then inhibit the protein translation machinery, causing the cell death. So this is the catalog number of the DT3C about our recombinant protein. And as a friendly reminder, attendees of this webinar can apply for a free trial of 100 microgram DT3C after you fill out a follow-up survey after the webinar. The follow-up survey will reach you via the email after the webinar. So contact us, get the free trial. And the recombinant protein, DT3C, has been verified by the SDS-phage and HPLC. So the purity is definitely higher than 90%. So the advantage of this recombinant protein DT3C is pretty clear. It can evaluate the internalization efficiency of antibody in vitro. And then the preparation between the antibody and the DT3C complex is simple. It's just room temperature, 30 minutes incubation, then you will get everything ready. And then the antibody can come from multiple sources, such as the mice, the rabbit, and then the goat. And then you can just use the MTT cell proliferation, as I showed here, to assess the internalization efficiency. And MTT is not the only choice. You can also use the WST1, the CTG, and or some other assays to assess the efficiency. And more importantly, according to the experimental result, our DT3C behaves better, have a higher efficiency than the MAB-ZAP. And the MAB-ZAP is a previous internalization efficiency tool. So we have a better performance about the DT3C. So another case about the DT3C is that you can use it to do a direct discovery of the potent ADC-compatible antibody in vitro. So here I showed a screening pipeline to describe how you do it. So first you do a immunization, and then you have the cell fusion of the B cells with the memory cells, and then you have a construction of the hybridomal library. So with the supernatant from the hybridomal library, you can incubate them with the supernatant and then DT3C protein. Then finally, you will get a monoclonal antibody with excellent internalization capacities. So just a simple screening pipeline, you will get what you want. So by now, we already have some understanding about the ADCs, but there's still room for the improvement. I want to share some of ideas that might be useful for next generation of ADC. So the first is about internalization ADC. Instead of using the internalizing mechanism, what about the non-internalizing mechanisms? Because we know that there were some limitations in the solid tumor because of the poor penetration and the lack of suitable internalizing antigen targets. We know that there are multitude of extracellular markers from the tumor microenvironment as well as the cancer stroma. So one example about this is the galectin-3 binding protein. So this protein is abundant and then continuously secreted from the most cancers, making it an attractive target for the ADCs. And also comparing to the antibody, the small molecules, the diffusion rate is greater than it. So it can increase the bystander effect. And then actually in general, for solid tumors with these strategies, with the increased bystander effect and the tumor penetration, people found that the treatment may be better with the solid tumor. However, if you choose the non-internalization ADC strategy, then the chemistry of the ADC linkers may be essential to be optimized. So find a optimal one, then try the non-internalization ADC may be useful for the next generation. And then the next part is about the optimization of the antibody. So instead of using the single monoclonal antibody, what about the bispecific antibody? Because for this one, you can choose 2 different antigen binding sites. And then this will result in improved therapeutic efficacy comparing to the previous one. And then you can see that the enhancement, the treatment can also be enhanced. And actually there's already a clinical trial called the DT2219 already in process. It targets the CD19 and the CD22 simultaneously to increase the ADC internalization. And then another strategy is about the antibody size. So instead of using the whole antibody, what about the smaller format, like the single chain variable fragment format? And also to make the antibody better, we can also choose the immunogenetic cell death, like triad. So I know I already talked about some of the advancement in the linker and the conjugation technology, but I still want to say that the ADC drugs, they are heterogeneous mixtures. So optimization of the coupling technology can improve them to be homogeneity. So to have the optimal efficacy as well as the optimal safety. Then overall, you will have less variabilities in the dose studies. And then the therapeutic windows can be also increased. So considering the optimal efficacy and the safety, we cannot ignore the pharmacodynamics as well as the pharmacokinetics. So the ADC drugs have a very complex pharmacokinetic profiles due to the heterogeneous mixture of the product. So it's a kind of like a population that you have the naked antibody and the conjugate antibody together. So it's challenging to establish PK and the PD models to describe the clinical characteristics of the ADC. And then also it's hard to design for the new ADC drugs. And another feature about ADC I want to say is that the hydrophobicity balance. So think about it. If the ADC is too hydrophobic, they will aggregate, and then they will be cleaned up too quickly. So you can imagine that the efficacy will also be decreased. And then the last thing about the optimization is about the payload. So people already try some new type of payload in the clinical trials, such as the RNA polymerase inhibitor. So for this type, they kill the tumor cells by inhibiting the transcription, and then which is required by all cells. So with the new trial of the new type of the payload, we probably will have some ideas about how to avoid the drug resistant. And then the last thing is that the same as the bispecific antibody, what about the dial payload ADC? So using two different payloads, we probably can increase the potency of the ADC a lot. So overall, we see that during the development of the ADC, the therapeutic potential of the ADC is highlighted by the explanation of the clinical indications. And then we see that there's a shift from the hematological malignancy to the solid tumors. And then as we all realized, it's important to always keep in mind that ADC drugs are whole. So choosing either side will affect the other. There's always a balance that we want to get to develop a successful ADC. So here, please let me introduce my company. The Kusel Bio is a manufacturer, and we offer a wide range of products for research purpose. And we are now expanding into the pharmaceutical industry. We have a vast selection of the products in the field, such as the cancer research, cell biology, immunology, neuroscience, and epigenetics. And we believe that the biotechnology makes life better. So our products are exported to more than 80 countries, including Canada, the US, Germany, and the Japan and Korea. And we have more than 11,000 kinds of products developed, covering both the conventional product and as well as the unique products. We will provide our customers with the proteins that they cannot be found elsewhere, and then we will try our best to meet our customers' various demands. And then here, this table summarizes some of our best-selling proteins. They all have biological activities, and then they are validated on multiple platforms. Most of them, they are very popular. They are drug target proteins. And then except the proteins, we also have antibody products. We have a wide selection of the antibody products for multiple applications, such as the ELISA, the Western blot, and the immunoprecipitation, as well as the flow cytometry. The quality of our antibody are all verified and supported by the experimental data.